Good morning, my dear students. Again, I am back with a very interesting topic. These are the cell structures. Okay, so in the cell structures, we will see about what is a cell, what is the cell theory, what about the microscopes, and etc. Okay, history of the cell. Some points about history of the cell. Okay, in 1665, Robert Hooke observed the cork cells or the dead cells and named them as the cell. In 1675, Anton van Leeuwenhoek was the first to describe the living cell seen through a simple microscope. Compound light microscope, it contains series of lenses that magnify the objects. In 1665, Robert Hooke observed the cork cells, dead cells, named the cells. The cell theory was given by Sheldon, Swan, and Virchow. Again, I am repeating history of the cell. 1665, Robert Hooke observed the cork cells. These were the dead cells, and he named the topic as the cell, the title. In 1675, Anton van Leeuwenhoek was the first to describe the living cell seen through a simple microscope. Compound light microscope, these contain a series of lenses that magnify the objects. In 1665, Robert Hooke observed the cork cells or the dead cells and named the topic as the cell. Sheldon, Swan, and Virchow were the authors of the cell theory. Cell theory. These are the basic points. Kindly noted down because these are very much important. The cell is the basic functional and structural unit of all forms of living life. All the living things are composed of cells, cell parts, or the cell products. All cells come from the pre-existing cells. Cell. Sorry, my dear friends, for the disruption. Okay, now coming on to the cell theory, this is a very interesting and important topic. Okay, the cell is the basic functional and structural unit of all forms of living forms of life. All the living things are composed of cells, cell parts, or the cell products. The cell parts are the nucleus, nucleolus, mitochondria, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, Golgi body, etc. All cells come from the pre-existing cells. Electron microscope, these were developed in the 1940s. These aim a beam of electrons through magnetic field then over the surface of the specimen. These uses the electromagnets. Most specimens must be dead and in a vacuum environment. These projects, the projections are onto a screen or on a monitor. In 1940s, the electron microscope were developed. These aim a beam of electrons through a magnetic field, then over the surface of the specimen. These uses electromagnets. Most specimens must be dead and in the vacuum. These projects onto a screen or a monitor. Electron microscope, various types. Scanning electron microscope or the ACM. These are ion coat. There, these contain ion coated surface. They show a 3D picture. Transmission electron microscope. These are slice stained with metal ions. Scanning tunneling electron microscope. This creates a 3D surface image of a living cell. The various types of electron microscopes are scanning electron microscope. These are ion coated surfaces shows a 3D picture. Transmission electron microscope. These are slice stained with metal ions. Scanning tunneling electron microscope. It creates a 3D surface image of a living cell. Eukaryote cell and a prokaryote cell, or what are the basic differences? Two cell types, prokaryote cells, these have no membrane-bound nucleus nor membrane-bound organelles. Most are single-celled. Examples are bacteria and cyanobacteria. Again, I am repeating prokaryote. There are two cell types. Prokaryotic cells, these have no membrane-bound nucleus nor membrane-bound organelles. Most are single-celled. Examples are bacteria and cyanobacteria. Two cell types again. Eukaryotic cells have a membrane-bound nucleus and membrane-bound organelles, either single-celled or multicellular. Organelles are separated by membranes, so several processes can be going on at once. Lin-Margulis hypothesis suggests that eukaryote cells come from prokaryote cells. Again, I am repeating, eukaryotic cells have a membrane-bound nucleus and membrane-bound organelles, either single-celled or multicellular. Organelles are separated by membranes, so several processes can be going on at once. Lin-Margulis hypothesis suggests that eukaryote cells come from prokaryote cells. Plasma membrane. This phospholipid bilayer, one very important thing to be noted in the plasma membrane is that this phospholipid bilayer membrane acts as a selectively permeable gateway for the passage of substances into and out of the cell. Boundary between cell and the environment controls materials that enter and exit the cell. Example, oxygen and nutrients come inside and waste materials they go outside. The membrane also contains proteins embedded within its phospholipid layers that act to encourage molecular movement or as recognition molecules. Now coming on to the cell structures. Cytoplasm, jelly-like substance that surrounds the organelles, makes up half of the volume of the animal cells. Many important chemical reactions occur here. In this diagram, you can see ribosome, cell membrane, mitochondria, nucleus, nucleolus, endoplasmic reticulum, Golgi body, Golgi apparatus, etc. Organelles, the nucleus. The nucleus is the command center of the cell, surrounded by a four nuclear envelope, double membrane, four layers thick. Pores in the nuclear envelope allow chemical messages to move in and out of the nucleus. Contains DNA made up of long strands called chromatin threads. The nucleolus inside the nucleus produces the ribosomes. This point is very much important, my dear friends. The nucleolus inside the nucleus produces the ribosomes. Ribosomes, the protein synthesis factories, it makes enzymes and the proteins composed of RNA, non-membrane structures. These are directed by the deoxyribonucleic acid or the DNA. Mitochondria, known as the powerhouse of the cell. This is very much important and very much important again from the MCQ point of view. Site of aerobic respiration. These organelles capture the glucose and use oxygen to release energy for the cell. Holdings within the mitochondria are called cristae. The reaction here is very much important. Oxygen plus C6H12O6 plus mitochondria gives energy in the form of ATP plus H2O plus CO2. 2. You can have a short note on mitochondria in your final exam, so kindly study it well. Endoplasmic reticulum, bilayer of lipids used for transport within the cell. Two types. Smooth endoplasmic reticulum does not have ribosomes attached and are known to produce the lipids. Rough endoplasmic reticulum have ribosomes attached and are known for both protein synthesis and the lipid synthesis and the transport. Golgi apparatus, this is known as the packaging factory, a series of flattened, closely stacked membranous sacs near the nucleus that coats the enzymes. Lysosomes, these are the stomachs of the cells or the bud off from the Golgi bodies as the cell needs. Lysosomes are the suicidal bags or the suicidal factories, digest one-out cell parts, food particles, and viruses or bacteria. Centrioles are only found in the animal cells. These are two small bundles of cytoskeleton arranged at right angles to one another and surrounded by a dense cytoplasm. Centrioles aid in cell division in the animal cells. The very characteristic feature to be noted here of the animal cells is that only animal cells have cell wall, whereas the human cells have the cell membrane. Lysosome, these are suicidal sacs which are filled with the digestive enzymes used to break down molecules within the cell. As complex molecules enter the cell, lysosomes surround food and digest it. Vacuoles. Vacuoles are storage vessels found within the cell. Plant cells usually have large vacuoles, while animals have vacuoles that are small or even absent. Vacuoles can store food, water, or waste products. A contractile vacuole is a specialized vacuole to remove excess water from unicellular aquatic organisms. Understood, my dear friends, up till here? Chloroplasts, only found in the plant cells filled with the green pigment chlorophyll. These organelles function to transform light energy into stored energy, that is glucose and starch. Grana, stacked membranous sac stroma, fluid that surrounds grana. Cell walls are only found in the plants are protective dead layers of thick cellulose, bulk of fiber in diet. In fungi, cell walls are made out of a carbohydrate called chitin. Cell wall and the cell membrane, as you can see. Movement and support, cytoskeleton, fibrous provides support for the organelles, maintains the cell shape composed of microtubules and microfilament, assist organelles to move from place to place. Cilia and flagella. Cilia, short, numerous, hair-like bit in a wave, example lungs. Flagella, longer, 1 to 2 per cell, move in a weave-like motion, example sperm cells. Single-celled organisms usually depend on cilia or flagella to move. Multicellular organisms use cilia on the cells to move fluids over the surface of tissues. Organization, cells, tissue, organs, organ systems, and then organism, tissue. A group of cells organized together, linked at the cell junctions. Examples: muscle, nerve, brain, bone, blood, or root tissue. Organs, group of two or more tissues functioning together. Example: stomach, heart, or a leaf. Organ system, group of organs working together to carry out major life functions, example nervous system or flower. Organism, group of organ systems working together, examples human, dog, cat, plant. Okay, understood, my dear friends? Why can't cells grow to a enormous size? The ability of cell membrane to absorb and release substances prevents them from growing large. One way to explain this is to say the surface area to volume ratio. Demonstrate that there is not enough surface area to absorb the nutrients quickly enough to keep cell alive. As it grows larger, thus the cell must divide. So this is all for the cell topic, my dear friends. I hope you have understood well. So